DNA fingerprinting is the process of determining an individual's DNA characteristics. DNA profiling, DNA testing, DNA analysis, genetic testing, genetic analysis, genetic fingerprinting, genetic profiling are some of the popular names used for DNA fingerprinting. In all the body parts, in all tissues, our DNA is the same except in case of germ cells. Even after the death of an individual, it remains the same. It can remain stable even after thousands of years. On average, about 99.9% of the DNA between two humans is the same. The other 0.1% is what makes each one of us unique. The majority of human genetic variation resides in non-protein coding regions of the genome. This polymorphism occurs due to specific codes or regions which is present in the non-coding regions of the genome. These short and varied sequences or regions are called variable number tandem repeats (VNTRs) or mini-satellites and short tandem repeats (STRs) or microsatellites. VNTRs or mini-satellites are 10 to 100 base pairs in length. STRs or microsatellites are 2 to 4 base pairs in length. The frequency and position of these repeats vary greatly from one individual to the other. These repetitive nature and polymorphic properties of junk DNA are utilized to perform DNA fingerprinting. The number of repeats and the sequence structure of these regions vary between individuals, and based on this, the entire DNA profile is made. The process of DNA fingerprinting was invented by Sir Alex Jeffreys at the University of Leicester in 1985. Professor Jeffreys' research at Leicester focused on exploring human DNA diversity and the mutation processes that create this diversity. He was studying how inherited illnesses pass through families. As a part of this work, Jeffreys had to look for patterns in the repeated DNA segments carried by all humans. Jeffreys noticed that certain sequences, highly variable sequences of DNA that present in the non-coding regions are repeated within the genes. He recognized that each individual has a unique pattern of mini-satellites. Jeffreys says he had a eureka moment in his lab in Leicester after looking at the X-ray film image of a DNA experiment on 10 September 1984, which unexpectedly showed both similarities and differences between the DNA of different members of his technician's family. Finally, Alec realized he had produced the first DNA fingerprints. In India, DNA fingerprinting was first started by Dr. V.K. Kashyap and Dr. Lalji Singh. Dr. Lalji Singh is popularly known as the father of Indian DNA fingerprinting. During 1970s, while studying the evolution of sex chromosomes in a species of Indian snake, the banded krait, Dr. Lalji Singh and his colleagues identified certain DNA sequences, highly conserved DNA sequences in banded krait and also in other vertebrates. They named it as banded krait minor or BKM sequences. These BKM sequences were conserved across various species and were found to be polymorphic among humans. In 1987 through 1988, while working in the CCMP, Singh established that this BKM-derived probe could be used to generate individual-specific DNA fingerprints of humans for forensic investigation. In 1988, he used that probe for the first time to solve a case of parentage dispute in India. DNA fingerprinting methods. Method 1, restriction fragment length polymorphism or RFLP based VNTR analysis. This was the first method developed by Jeffreys for DNA fingerprinting. Step 1 is the extraction of DNA from the cells or sample. DNA can be acquired from any bodily sample. Buccal smear, salivation, blood, amniotic liquid, chorionic villi, skin, hair, body liquid, and different tissues are significant kinds of samples utilized. DNA can be extracted with different methods like CTAB DNA extraction methods, proteinase K DNA extraction methods, and phenol chloroform DNA extraction method, or maybe with any DNA extraction kit. Step 2 is digesting the DNA with restriction endonuclease enzymes. DNA is collected from the cells or samples and cut into pieces at specific points using restriction endonuclease enzymes. Step 3 is the separation of DNA on a gel. Here, agarose gel electrophoresis is used. The enzyme produces DNA fragments of varying length were sorted by placing them on a gel and subjecting the gel to an electric current. The electric current pulls the negatively charged DNA through the gel. The shorter pieces of DNA moves through the gel fastest and the longer pieces of DNA move slower. As a result, by the time the electric current was switched off, the DNA pieces had been separated in order of size. Step 4 is the transfer of these separated DNA fragments onto a synthetic membrane. Here for transfer we use Southern blotting apparatus. In Southern blotting, the DNA pieces are transferred to nitrocellulose paper or a nylon membrane by placing it over the top of the gel and placing absorbent paper over the top of the membrane. Step 5 is hybridizing the DNA fragments using labeled VNTR probes. Here, the synthetic membrane was incubated with the probes tagged with chromogenic or radioactive or fluorescent dye. This helps in detecting the matched DNA visually. The probes only attach to the pieces of DNA that they are complementary to. In this case, they attach to the mini satellites in the genome. Step 6 is the analysis of these hybrid fragments using autoradiography. After probing, the excess probes are washed away and the resulting membrane is visualized using autoradiography. The X-ray film thus developed shows the hybridization pattern. The DNA sample thus hybridized with specific probe can be seen and studied. This hybridization pattern is called DNA fingerprint. This RFLP based VNTR analysis is considered to be more accurate because of the use of large amount of fresh DNA sample and there is no amplification contamination, but it requires longer time period to complete the analysis and is costly. Jeffrey's All method has been supplanted by another approaches that use PCR-based microsatellites or polymerase chain reaction-based microsatellite analysis. Microsatellites are the shorter repeated units which are shorter than mini satellites.